Hi, my name is Debbie Figarelli. I'm the DNA Technical Leader at the National Forensic Science Technology Center and your instructor for today's class. The training program that you are about to work through is based on the material prepared by the NFSTC and funded by the National Institute of Justice. The topic is Population Genetics and Statistics. Upon successful completion of the course, you will be able to 1. Describe the basic population genetic principles that influence allele frequencies. 2. Describe the statistical principles that govern interpretation of genetic population data. And 3. Apply these genetic and statistical principles to interpretation of DNA test results. The training is presented in a modular fashion with a self-contained podcast covering each objective. Use the play and pause controls to navigate through the program. There is also an integrated web version that includes interactive hands-on sessions and a final assessment that can be used to demonstrate competency in the subject. The course can be accessed at projects.nfstc.org/popgen. The material is also available in a different form in the DNA Analyst training class on dna.gov. Finally, you should have access to a copy of the National Research Council report, the Evaluation of Forensic DNA Evidence, commonly referred to as the NRC II report, and available through books.nap.edu. This is the first module, and it addresses learning objective number 1, describe the basic population genetic principles that influence allele frequencies. The module learning objectives are 1. Define the Hardy-Weinberg principle. 2. Define Hardy-Weinberg equilibrium and describe the factors that influence it. 3. Calculate the allele frequencies from sample genotype data. And 4. Explain the chi-square test as it applies to Hardy-Weinberg equilibrium. I will take you through the material using slides and also some video clips from a workshop presented for NFSTC by Dr. Greg LaBerge, Director of the Denver Police Department Crime Laboratory. Let's look at the basics of population genetics. In order for us to be able to comment on the frequency of an allele or a complete DNA profile, we need information about the relevant population. This requires some statistical manipulation of data that we will deal with here, even though there is a separate statistics module. Some simple statistical techniques are required so that we can use data from samples to make inferences about populations. We can measure the frequency of an allele in the sample and then use this as the basis to estimate the frequency of the allele in the population as a whole. Statistics allow us to understand how good the sample value is as an estimator of the population value. This is important because we are asked to explain our results in terms of the frequency of the alleles that we have found in the population of interest. We do this using databases constructed from samples of relevant populations, such as Caucasians, African Americans, Hispanics, and Asians. We have to use samples because it is impossible to access the whole population. The size of the sample will affect the accuracy of our estimates. A minimum sample of 200 has become the de facto standard for database size. Factors like race and geographical isolation can influence the frequency of alleles in a population, and the sample must be well characterized. The underlying principle used by population geneticists in characterizing populations is the Hardy-Weinberg principle. The Hardy-Weinberg principle states that in a large, randomly breeding population, allelic frequencies will remain the same from generation to generation, assuming that there is no mutation, gene migration, selection, or genetic drift. For a gene locus with two alleles, P and Q, this can be expressed as P squared plus 2PQ plus Q squared equals 1. When a population meets all of the Hardy-Weinberg conditions, it is said to be in Hardy-Weinberg equilibrium. Here is Dr. LaBerge talking about the Hardy-Weinberg principle and genetic drift.
What is genetic drift? You've heard that before. What is it? It's just, what's the word stochastic mean? Have you heard that word before? Stochastic effect. You probably heard it with respect to PCR. If you have 4 red socks and 4 black socks in a drawer, and you randomly go in and pick one, what's the chance of getting one or the other? 50%, right? If you have 1 and 1, what's the chances? Still 50%, but if you're picking out one by chance and copying it, if you only have 2 to choose from, the chances are still 50%, but if you start copying that black one, it's going to appear like there's more black socks than there are red socks because you just by chance chose that one black sock. But if you had 300,000 of each in the drawer and you picked it, it's still, you know, remember PCR, you're picking a bunch at the same time is the analogy. But the point being that if you pick one and it's very, very little to start, you might get an incorrect representation of how many are actually, what the proportion between them actually are. And that is, in this sense, the heterozygotes, right? You might have one peak way up here and another one down here, which would falsely indicate there's more of this than this. It's just because you weren't choosing very many to choose from in the beginning stages of the PCR. Stochastic effect. Stochastic meaning just by chance, fluctuation. It's the same thing with genetic drift. Alleles become fixed in populations. When I say fixed, I mean common enough that they're seen in a lot of people, that is more than 1%. Sometimes just by genetic drift, by chance.
The Hardy-Weinberg principle was first published in 1908 and defines the relationship between genotype and allele frequencies at a locus. For two alleles, P and Q at a given locus, the frequency in the population of people homozygous for P will be P squared, and the frequency of heterozygotes will be 2pq. We can therefore use a simple counting of homozygotes and heterozygotes in a sample of the population to calculate the allele frequencies. The degree of genetic variability across loci can be indicated by measuring the amount of heterozygosity. For a locus with k alleles, the number of genotypes is given by the equation k times k plus 1 over 2. The expected number of heterozygous genotypes is given by k times k minus 1 over 2. We can count the number of heterozygotes in a sample and compare it to the expected heterozygosity. The comparison gives us important information about the population dynamics. Calculations based on allele frequency data are important in measuring the degrees to which populations are in Hardy-Weinberg equilibrium. Human populations generally do not meet all of the exact conditions of Hardy-Weinberg equilibrium. Their allele frequencies change from generation to generation, and the population evolves. How far a population deviates from Hardy-Weinberg equilibrium can be measured using the goodness of fit or the chi-squared test. The chi-squared statistic is calculated by listing the observed and the expected frequency of each allele and the difference between them, and squaring the values, which converts each to a positive number. The squares are then converted to a proportion of the expected, O minus E squared divided by E, and then summed for all alleles. To determine what this chi-squared value means, we look up the calculated chi-square value in a chi-squared distribution table. Note that the chi-squared distribution table uses something called degrees of freedom. Degrees of freedom is a complex issue, but we could look at this in simple terms. If we have frequencies for 3 genotypes that are truly representative of the population, then no matter what values we have for 2 of them, the frequency of the third must not be significantly different from what is required to fit the population. Hence, in using the chi-squared statistic, we have number of genotypes minus 1 degree of freedom. To illustrate how the Hardy-Weinberg principle works, let us consider the MN blood group. Humans inherit either an M or the N antigen, which is determined by the different alleles of the same gene. If we let the frequency of allele M equal P and the frequency of the other allele N equal Q, then the next generation's genotype will occur as follows. The frequency of the homozygous MM genotype equals P squared. The frequency of the heterozygous MN genotype equals 2pq, and the frequency of the homozygous NN genotype equals Q squared. We can take a sample of the population and count the number of people with each genotype. For example, a sample of 5,000 from Forensic Town, USA has 1,460 individuals of type MM, which is 29.2% of the population, 2,550 individuals of type MN, which is 51% of the population, and 990 individuals of type NN, which is 19.8% of the population. If we apply the Hardy-Weinberg equation, p squared plus 2pq plus q squared equals 1, we can calculate the allele frequencies. P equals 1 minus q, where p represents the frequency of the M allele and q represents the frequency of the N allele. So the frequency of M is p squared plus 1/2 2pq, which equals 0.547, and the frequency of the N allele is 1 minus p, which equals 0.453. We can now calculate our expected genotype frequencies. MM equals p squared, or 1,496 individuals. MN equals 2pq, or 2,478 individuals. NN equals q squared, or 1,026 individuals. Mathematically, the chi-squared test is the sum of the observed value minus the expected value squared divided by the expected value. Applying the above data to the chi-squared test is calculated using the O, or observed value, and the E, or expected value, to give a final chi-squared value of 4.221. Since we have 3 genotypes, we have 3 minus 1, or 2 degrees of freedom. Looking across the distribution table for 2 degrees of freedom, we find our chi-squared value of 4.221 is less than the 5.991, the value required to satisfy the hypothesis that the differences in the observed and the expected data did not arise by chance. Since the chi-squared value falls below the 5% significance cutoff, we can conclude that the forensics town population does not differ significantly from what we would expect for Hardy-Weinberg equilibrium of the MN blood group. Things that can prevent a population from attaining Hardy-Weinberg equilibrium include genetic drift, caused by factors such as selection, mutation, and migration. Here is a video clip of Dr. LeBerge talking about random mating.
Let's talk a little bit, and I talked a little bit earlier about random mating, but it's a key assumption under which you guys are operating. When you calculate with a product rule calculation in PopStats, by saying p squared and 2pq, you are assuming Hardy-Weinberg equilibrium. Okay? One of the key components of that is random mating. So random, what is random first? When someone says, I chose these people for my clinical study at random. Okay, I've been in a medical school for a long time. We've done a lot of medical studies. I put things up in the elevator sometimes. We want people to come in and give us their blood, okay? And in a medical school, who do you think's in a medical school? Medical students, graduate students. Anytime you go to select anybody, there's going to be some level of criteria, right? Like, so if I was doing a population study in diabetics, you know there's a big difference in diabetes between African Americans and Caucasians, right? If I was doing a study in diabetics, wouldn't I want the control group to be of the same racial classification as the test group or the question group? Yeah. So I'd have to impose a selection. So it's not random, right? Is there such a thing as random? No, there isn't. But the caveat to all this is random is only with respect to the parameter being studied. Remember that. Because if I'm studying certain receptor levels on the islet cells for diabetics, certain levels of the presence of a receptor, well, I don't have any preconceived knowledge of how many receptors our cameraman is going to have on his islet cells. If I select him as a control for a study, the thing I'm studying is random with respect to that. So in these genetic studies, when you go out and you select a group to genotype to count their alleles, you don't know their genotypes a priori. You know that they belong to some kind of a group. In the clinical study, I wouldn't know how many receptors they had on their islet cells unless they had diabetes. So we'd have the diabetes group, and then the control group was the only way it's random is you don't have diabetes, right? And you're of a certain racial category because we know there are differences in the prevalence of diabetes between the major racial categories. So remember that when the defense attorney says to you, were these people really selected randomly? They got them from blood banks. Well, that's random with respect to the STR. You didn't pick them for that. Okay, the other thing that comes into random is the mating part. And I talked to you a little bit in the very beginning of the course this morning about mating. It's based on continental proximity. The point being is that you don't choose your mate based on your genotypes. And it came up in our court case, and the redirect by the DA was, so when you chose your wife, did you know her VWA type and her FGA type? I said, no, I didn't. And he says, and you're the one person that could have, right? I said, yeah, I probably could have. But she wouldn't let me, I said. We got a big laugh, and the jury laughed, and everybody laughed. Well, all the defense attorney knew was the random mating. He just wanted to play around with the word random mating. That's why I'm trying to really nail it down for you. It's random with respect to the parameter being studied, and the mating may not be random with respect to race and geographical proximity, but it sure is with respect to the STR genotypes, right? Okay.
To summarize what Dr. LeBerge said, in addition to changes caused by mutation, allele frequencies will change if migration occurs into or away from the population. The effect of migration on Hardy-Weinberg equilibrium is dependent on the differences in allele frequencies between the donor and the recipient populations. Gene flow is another way to introduce genetic variability to a population. Similar to migration, it occurs when members of one gene pool mate with members of another gene pool, which can lead to an alteration of the allele frequencies. Inbreeding is the mating of related individuals and can alter gene frequency in a population. When related individuals mate, the child can inherit identical copies of the gene through both parental lineages, resulting in an increase in homozygosity at any given locus. The degree of a relationship between two persons can be measured using the inbreeding coefficient, F. With inbreeding, the expected heterozygosity is reduced by a fraction, F, and that of homozygotes is increased. For the purposes of this training, theta and F will be regarded as being the same. The difference between populations that are diverging by drift is often characterized by theta, or F. It is considered a co-ancestry coefficient. For example, the inbreeding coefficient F between a parent-child is 1/4, for siblings is 1/4, and for first cousins is 1/16. We will end this segment with a video clip of Dr. LeBerge discussing inbreeding as described in the second NRC report.
The main thing you want to do is understand NRC II's recommendations. And that's what the field wants you to refer to until there's a new document that comes out, which I doubt really there's a need for. And this document was motivated out of the fact that there were some errors in terms of thinking in NRC I, and that population substructure needed to be addressed in the forensic community because mainly the defense was bringing it up in cases. And they were bringing it up in the completely wrong context. And that wrong context was, well, my suspect's from an island in the Caribbean. The question that you're trying to answer with the random match probability is, what's the probability of seeing that profile by chance in the general population? It has nothing to do with the suspect. It's irrelevant. But the defense was making that claim. So part of the response came in this document. There are a number of recommendations. The one that affects what we do with the random match probability is using a correction factor of what they're calling theta. It's also called, in my language, FST, or the amount of inbreeding. It's a measure of inbreeding. How much relative, or generally inbreeding is thought to be first cousins or less, like first cousins or brothers and sisters and things like that. Generally first cousins. And the number that they tell you to use is 0.01, right? So what is that? Theta equals 0.01. What does that mean? It's saying 1% of the population has some level of inbreeding. There's a 1% of the people are inbred. That's a lot. On an isolated or native population, 0.03 is what they recommend, or 3%. That's a lot too. Because I'm going to show you the real data now. Okay? When you look at the databases in CODIS, they have different populations that they've looked at. I think that paper was 2000, 2001, 41 populations examined. Yeah. 11 African American, 9 Caucasian, 8 Hispanic, 6 Asian, and 7 Native American populations. When you look at the inbreeding coefficient, FST, also called theta. Theta is just something it's assigned to that. Well, look in Caucasians for these STR markers and look what the actual level of calculated inbreeding is. And how do you know something's inbred? What would you expect to see at a locus if there was a lot of inbreeding? Increased homozygosity, right? Okay, well look at it. It's basically 0. It's negative 0.0005. In African Americans, 0.0006. 0.01 is several magnitudes more common than the actual numbers. Hispanic, 0.0021, Asian and Native American. Native American is really close to 0.03, isn't it?
Upon successful completion of this course, you should be able to answer these review questions. Number 1, a locus with 4 alleles has how many genotypes and how many heterozygotes? Number 2, Calculate the genotype frequencies for a locus with two alleles, K and L, where the frequency of K is 0.32. Number 3, for the above locus, calculations give a chi-squared value of 5.195. Is the population in Hardy-Weinberg equilibrium? Number 4, name 3 factors that can affect whether or not a population is within Hardy-Weinberg equilibrium. Number 5, what is the minimum size for a population database? And last, number 6, name 4 populations that are represented in CODIS. The class is continued in the next podcast on statistics. Please visit projects.nfstc.org/popgen to view the next video. And thank you for joining me today.